Hello everyone, I'm MS Shweta from 2nd MSc Microbiology. Today I'm going to present a topic, industrially produced enzymes by microorganisms. Enzymes. Enzymes are biomolecules that catalyze chemical reactions. Nearly all enzymes reproteins. However, certain RNA molecules can be effective and catalyzed too. The RNA molecules is known as coenzymes. In enzymatic reactions, the molecule at the beginning of the process are called substrate. Enzymes converts them into different molecules called products. Industrial enzymes are these which are used for industrial production, and also these are produced in industry. Example, amylase, protease and pectinase are the examples of enzymes. Amylase. Amylase is an enzyme that catalyzes the hydrolysis of starch to sugar. Amylases are the large group of enzymes that are categorized their ability to hydrolyze 1,4 glucosidic linkage in polysaccharides. Example, starch and glucogen. Amylase plays the most important role in food technology, such as bread making and beverage making. Amylase are classified into two subgroups. There are alpha amylases and beta amylases. Microorganisms used for amylase production, such as bacterial strains and fungal strains. In bacterial strains, most are Bacillus species, such as Bacillus subtilis, Bacillus menstricus, Bacillus vulgaricus, Bacillus megatherium, and also a Bacillus cereus. Amylases from Rhodothermus marinus, Cornybacterium gigantea, and Chromohalobacter species. Some lactospecies bacillus can also produce enzymes. In halophilic strains such as Haloharicula hispanica, Halobacillus species, Chromohalobacter species, Bacillus dipsoriae, and Halomonas merdenia. These are the bacterial strains used for the production of amylase, and few fungal strains producing amylase such as Aspergillus oryzae, Aspergillus niger, Aspergillus fumigatus, and Aspergillus flavus, as well as penicillium species such as Penicillium bruneum, Penicillium expansion, Penicillium chrysogenum, and Penicillium olsoni. Streptomyces, rhamnosus, Thermomyces, Landraneasis, Cryptococcus flavus, Thermomonospora curvata, and Mucus species can produce amylase. Bacterial amylase produced by the submerged fermentation. Submerged fermentation is a method of cultivating microorganisms in a liquid nutrient broth. For example, industrial production, this implies growing the selected microorganisms in a closed vessel. And also is a process involving the development of microorganisms in a liquid broth. Fermentation medium. Fermentation mediums are soya bean meal 1.85%, brewer's yeast fraction 1.50%, distillers dried solubles 0.76%, NZ-amine 0.65%, lactose 4.75%, MgSO4 7H2O is 0.04%, antifoam 0.05%, water 90.40 liters. Fungal strains can produce amylase by submerged fermentation. Wheat bran was used as a substrate. Aeration and agitation is a problem due to the high viscosity. Amylase synthesis is inhibited when medium contains glucose. Fermentation medium of fungal strains such as cornstarch 24 gram per liter, corn steep liquor 36 gram per liter, KCl 0.2 gram per liter, Na2HPO4 47 gram per liter, calcium chloride 1 gram per liter, magnesium chloride 0.6 H2O 0. gram per liter. Fermentation condition. Temperature should maintain 30 degrees Celsius to 40 degrees Celsius and pH should be in neutral or close to neutral. pH below 6 results in denaturation of amylase. Calcium carbonate is used to maintain neutral pH. The amylase production begins when the bacterial count reaches 109 to 110 cells per ml after 10 to 20 hours and continues for 10 to 150 hours. Duration of fermentation should be maintained 4 to 6 days. Recovery of amylase. Separation of fungal mycelium is accomplished by filtrations of the refrigerated broth. For bacterial amylase, there is no need of filtration. The suspended particles present in the broth are removed with flocculating agents like calcium phosphate. The enzymes is precipitated in order to get high degree of purity by using acetone or alcohol or even inorganic salts like ammonium sulfate or sodium sulfate. Sometimes fractional precipitations of the enzymes is done to obtain it in the purest form. Application of amylase enzymes. Alpha and beta amylases are important in brewing beer and liquor made from sugars derived from starch. Amylases are used in bread making and to break down complexes sugar such as starch into simple sugars. Amylase also has a medical applications in the use of pancreatic enzymes replacement therapy. Bacillary amylase is also used in clothing and dishwater detergents to dissolve starches from fabrics and dishes. Second enzymes is pectinase. Pectinase is the collective term for a row of enzymes that are able to break down or transform pectins. Pectinase is a general form for enzymes such as pectylase, pectizymes, polyglucuronase, etc., commonly referred as pectin enzymes. Pectinase break down pectin, a polysaccharide that is found in the cell wall of plants. Pectinase is involved in the metabolism of the cell wall as well as the growth of the cell. Senescence is a biological aging, ripening of fruits, pathogenesis and abscission process, shedding of various parts. The basic structure of pectin is 1,4 linked glutaronic acid with up to 90% of its carboxyl group. Esterified with methanol. According to the Enzymes Commission and the International Union of Biochemistry, pectinase enzymes are classified under hydrolase group. The majority of commercial enzymes are obtained by employing the fungal cultures. Pectinases have been commercially prepared from the microorganisms, especially from the fungi, since 1970s. Microbes used for the pectinase production. Pectinase produced by various bacteria and fungi, although commercial microbial pectinase production utilizes species of Penicillium and Aspergillus. Microbial strains Aspergillus niger, Aspergillus japonicum, Bacillus subtilis, and Penicillium italicum are common pectinase producers. Fermentation condition. Solid or submerged fermentations process are used for the production of pectinase enzymes. Substrates used for the production of pectinase such as wheat bran, rice bran, potato starch, rice husk, 0.1% of NaH4NO3, NaH4H2, PO4 and magnesium sulfide. The enhancer used for the pectinase producers are 2% of sucrose and 2% of pectin. Inoculum used for the pectin production is spore of Aspergillus niger in 107 spores per gram of dry substrate. Aeration is necessary for the production. Temperature should maintain at 37 degrees Celsius and the pH should be at 3 to 4. The duration of fermentations are 60 to 80 hours. Recovery and purification of pectinase enzymes. Pectinase can be extracted from the cell and the medium, so can be recovered from both sources. In the process of harvesting, the mycelium is filtered or centrifuged and then dried and also grind, and the pectinase extracted with water. And the next step, recover aqueous portion, is concentrated by evaporations to yield the pectinase concentrate. As an alternate recovery producers, pectinase can be precipitated from the aqueous solutions by the addition of cold acetone or ethanol or isopropanol or ammonium sulfate. These are used to separate the precipitated from the aqueous solution. Applications of pectinase. Pectinases are widely used in the food industry to improve the cloud stability of fruit and vegetable nectars, and also for the production and clarifications of fruit juices for the ash removal from wines. Pectinases are also involved in the clarification of wine, oil extraction, removal of citric fruit peels, and decoming fibers. Pectinase usage accelerates tea fermentation and also destroy the foam forming property of instant tea powders by destroying pectins. Pectinase used in coffee fermentation to remove mucilaginous coat from coffee beans. Pectinase used in the extractions of pigments, food coloring from the plant-based raw materials. Pectinase used in the industrial manufacture of feeds. Also, pectinase used in retting, that means separation of fiber from the plant parts. And the next enzyme is protease. Protease represents a broad group of enzymes which break down or hydrolyze proteins or peptides. Complex mixtures of proteinases and peptidase are called proteases. Proteases act on the peptide bonds joining the adjacent amino acid residues in a protein molecule and cleave them, leading to the fermentation of shorter peptides and amino acids. Proteases occupy a central portion in commerce, accounting for nearly 65% of the global enzymes market. Proteases or hydrolytic enzymes are ubiquitous in nature and have been found in eukaryotes such as plants, animals, fungi, and also present in prokaryotic domains of bacteria and archaea. Even several viruses also known to encoded their own proteases. Cysteine proteases identified in adenovirus alpha virus. Serine proteases are reported in hepatitis C virus. Aspartic proteases found in HIV. Types of proteases. Proteases are classified into three subgroups. There are alkaline protease, acid protease, neutral protease. First, alkaline protease, that the pH range is to 9 to 11. Serine protease, cysteine protease, and substelline Carey-Berg. These are the most widely used detergent proteases. Acid proteases such as aspartic protease, pepsin, and retropepsin. Neutral proteases are neutrase, that means thermolysin. Alkaline protease. Alkaline protease are produced by some of the bacteria such as Bacillus species, Bacillus megaterium, Enterococcus aureus, Pseudomonas aeruginosa, Streptomyces griseus, and Streptomyces rectus. Proteases are mainly produced from Bacillus lanchinoformis. Some fungal species can also produce alkaline proteases such as Aspergillus niger, Aspergillus oryzae, Aspergillus flavus, Aspergillus souchei, and Chaetomium globosum. And the next one is acid proteases. Acid proteases are found in animal cells, yeast, and molds, but never in bacteria. Microbial renin-like enzymes are derived from the mucus species, Aspergillus species, and Rhizobus species because bacterial strains cannot produce acid proteases. Neutral proteases. can be produced by bacterial and also by fungal strains such as Bacillus subtilis, Bacillus cereus, Bacillus megatherium, Pseudomonas aeruginosa, and Streptomyces griseus. Some fungal species such as Aspergillus oryzae and Aspergillus zoae. Alkaline protease fermentation medium. Subtilisin Carisberg from Bacillus lentiformis is the most widely used detergent proteases obtained by the submerged culture method. In 1971, Japanese scientist Koki Orezlose first reported the production of alkali protease from bacteria. Fermentation medium used for the alkaline protease such as starch hydrolysate 50 gram per liter, soya bean meal for 20 gram per liter, casein 20 gram per liter, sodium phosphate 3.3 gram per liter. Fermentation condition. The temperature should maintain 30 degrees Celsius to 40 degrees Celsius and the pH is 7. Calcium carbonate is used to maintain the neutral pH. The acid protease production begins after 10 to 20 hours of microbial cultivation. Duration of fermentation should be 2 to 3 days. Yield 10% of initial protein content of the medium. Enzyme stability of the alkaline protease is stable in liquid preparation and the liquid preparation contains 2% of enzymes. Acid protease of the fermentation medium mostly produced by fungi through semi-solid culture or submerged culture. Normally, the fermentation mediums contains 60% of wheat bran with water. Fermentation medium used for the acid protease are starch 4 gram per liter, soybean meal 3 gram per liter, ground barley 10 gram per liter, calcium carbonate 0.5 gram per liter. Fermentation conditions are temperature 30 degrees Celsius, pH should be in 7, duration of the fermentation is 3 days. Yield 3200 oxalate units per gram of wheat bran in mucor fermentation and 10,000 to 1,50,000 oxalate unit per ml of fermentation medium in mucor fermentation. Recovery and purification of alkaline and acid protease. Different methods can be applied for the purification of enzymes like ultrafiltration, chromatographic technique, that means the ion exchange purification by treatment with activated charcoal and hydrogen peroxide. Application of protease. Alkaline proteases mainly used in detergent industry and leather industry. Acetic proteases used in soya sauce, protein hydrolysate, digestive acids and in production of seasoning material and clearing beer and fruit juices, improving the texture of flour paste and tendering the fibral muscles. Neutral proteases can also used in food industry and brewing industry. Some MCQs are asked in exams. Most food and microbial enzymes are destroyed at dash degree Celsius, 79.4 degrees Celsius, because most food and microbial enzymes are destroyed at 79.4 degrees Celsius because higher the optimal and the maximum temperature for growth, the greater resistance can kill the food and microbial enzymes. Second question, which of the following is produced with the combination of apoenzymes and coenzymes? Options are alloenzymes, enzyme substrate complex, prosthetic group, enzymes product complex. The answer is alloenzymes. Alloenzymes are catalytically active enzymes that are coupled with cofactor or prosthetic groups according to the definition of biochemistry. The apoenzymes is the protein portion of the enzymes that does not include the prosthetic group. Thank you for listening.